Hello, everyone. Thank you for joining our sweet sorghum as a biofuel crop and forage crop presentation. Uh, Four of us will be presenting a different aspect of this research. So, sweet sorghum is a C4 plant, as we know, so it can be more efficient in photosynthesis and is able to grow in on the soil which may not be that arable and needs less water and can do uh, production of many of the products with less inputs. We know about sweet sorghum history in Tennessee, that is a southern crop, and since 1950s, the small farmers have grown it, and they have been successful in producing syrups, which could have competition with other products available. However, we can also switch the same syrup into biofuel uh, applications through fermentation process. At the same time, uh, after extracting the juice from sweet sorghum stems, we can use the leftover biomass as forage or for the lignoxylic uh, fermentation process. So either way, it would be very profitable for small farmers, and since different feedstock can be supported on small scale too. In our greenhouse, we have tested four different varieties: Achituri, Dale, Dutch Local, and Topper 76. And all those four varieties are known to be uh, sweet, sweet sorghum varieties. Topper 766 and Dale are Tennessee locally grown varieties, and while Achituri and Dutch Local are foreign varieties whose seeds are available within USA. And all four varieties gave good sugar production, and after evaporation of the water contents, a fermentable level of syrup was produced. Achituri and Topper 76 are also better in silage, whereas Dale and Dutch Local have thicker stem, which are more feasible for sugar extraction. So I just mentioned earlier that syrup has from sweet sorghum has a long history within Tennessee, uh, and so it should be a very easy switch for small farmers to use it for biofuel production. When we want to increase the production rate of sweet sorghum, we have to employ genetic tools. And to start with, the breeders always goes for flowering parts of the plant, or here we can concentrate directly on pollen grains or the gametes. We know that pollen grains have very thick cell wall, which is known as axine, and also intine within. But if we can target an earlier stage, it will be behaving as natural protoplast, which is very similar to animal cell genetics uh, tools which we can employ. Single cell genetics or single gamete genetics is getting popular since we can use precise genetic tools for manipulation as well as analysis, which can help to us to improve the sweet sorghum in all the production traits we are targeting, and a small farmer might be looking for to improve. Now we'll switch to the three different student research projects. Miss uh, Gordon, Mr. Yahya, and Mr. Felds, who are doing BSc, Masters, and PhD theses or researches on this uh, Tennessee crop. So with that, I introduce Miss Gordon to introduce her research to you. Miss Gordon.
Hello, everybody. I am Trinity Gordon, and I'm a sophomore at Tennessee State University, majoring in agriculture with a concentration in biotechnology. And I will be discussing with you all today cell division and microsporogenesis. Okay, so first, I would like to discuss cell division for pollen development. And this overall process deals with chromosomes that are being duplicated, and each of them has two chromatids that are joined together. Now, in this process, there is meiosis 1 and meiosis 2, but we will just talk about meiosis. So for meiosis 1, there's chromosomes of each homologous pair that are being separated from one another. And with meiosis 2, there are chromatids of each chromosomes being separated from one another. So that's the difference between the two. So for meiosis, this process is where a single cell is being divided twice to produce 4 haploid cells that contain only half of the genetic material from the original cell. So there is 2 pairs of chromosomes in the first stage. There are 2 pairs of chromosomes that have not been duplicated yet, and 2 homologous pairs. That's the end product. And next, there are 4 chromosomes pairs that are duplicated, 8 chromatids, and 2 homologous pairs. And in meiosis 1, the homologous pairs are being separated from one another as well. So in the endomeiosis 1, there are 2 daughter cells that have been divided by the mother cell. And these cells are called gametes, and this makes it possible for organisms to reproduce. Okay, so now this is just an overview of FDR and SDR. And so FDR stands for first division restitution, while SDR stands for second division restitution. With both FDR and SDR, the end results are unreduced cells that are broken into 2 and produce 4 chromatids and 2 pairs of homologous chromosomes. And the only difference between FDR and SDR is that FDR has no meiosis 1 and SDR has no meiosis 2. Okay, so now I would like to talk about microsporogenesis. Now, for the main points of microsporogenesis is the formation of microspores inside the pollen sacs of plants. In a sense, it is similar to meiosis, in which the mother cells and the microspores divide into 4 haploid cells. And then there is further development after the tetrad formation. So yes, and this is just the overview of everything that has been said about microsporogenesis.
Okay, thank you again, Ms. Gordon, that she described her work regarding what she is studying in terms of uh, pollen grain development. And now Mr. Yahya is going to describe how he is correlating and harvesting and doing genetic analysis on these on the seed sorghum.
Yeah, hello, good afternoon, everybody. Yes, my name is Dilovan Yahya, first year master's student at Tennessee State University, studying molecular genetics and sweet sorghum. We're gonna basically be discussing about egg sign development and proper cell isolation procedures as it pertains to the cells of the sweet sorghum plant. Basically, and ideally, we have stages that we are able to isolate and extract the DNA in, which are the late unicellular and early unicellular stages, right before the seed becomes a mature pollen grain, basically. So out of the four varieties that Dr. Aziz had mentioned, we're going to talk about Dell and Topper. So basically, we have a germination process that basically gets the seeds, and then we plant them in pots in a greenhouse in or residence, depending on our circumstances at hand. We use particular measurement tools such as the MicroMaster to identify the spikelet diameter and lengths as it pertains to the panicle from the plant in itself. And then we're able to observe the flag leaf emergence between the periods of time where it first emerges to the period of time that we're able to harvest, giving us an ideal stages that we can clearly observe the proper stages for the microspores growth in itself in order to identify the early unicellular and the late unicellular stages for proper cell isolation. And then we're basically taking all that correlated data and configuring and basically confirming it within the growing days of the microspores all the way up until the 13th day. And we can actually identify the growth from the bottom of the panicle all the way to the top of the panicle, basically stating that, yes, the microspores are growing and maturing through time. And then basically we're doing a maceration process utilizing sorbitol solution where we're able to stabilize the cells in the solution. So basically what we're doing, we're taking the microspore and then we're retracting the anthers, and within the anthers you potentially have between 20,000 to 60,000 cells that you're able to isolate by itself, of free microspores that you're able to isolate basically. And then these are the steps that we take for the cell isolation procedure, where we take the plant being harvested and retract the microspore to retrieve the anther and place it upon the micromanipulator and the Nikon microscope in order to isolate the cells and place them into PCR tubes for further genetic analysis. And this is one of the instruments that we use for the capillary tubes in order to divide the elongated capillary tubes into two via the capillary puller. Another instrument we have is the Celtron micromanipulator, where you're able to isolate the cells individually, as small as they are, basically utilizing a hydraulic pump through the Celtron device in itself. And then we basically get to the amplification of the cells in itself, utilizing a single cell Repligy kit, where we're able to get multiple displacement amplification of high quality, meaning that we get more DNA and less protein content by utilizing a polymerase Phi-29 that can navigate the structures in a proper fashion. And this is one of the charts that basically just verifies that the Repligy kit is doing its job in quality, in a sense of having a high nucleic acid quantity compared to a low protein content within the cell isolation procedure, utilizing a nanodrop device, a Viva spectrophotometer.
Thank you. Yeah, thank you, Mr. Yajas. So we understand that the sorghum microspore gave ample DNA for genetic analysis. Now, Mr. Aaron Feltz is going to present. He's a new PhD student and how he is interested in sweet sorghum genetic manipulations. Aaron?
Thank you, sir. First, I would just like to thank you, Dr. Aziz, for making all of this possible. Because really, without you and your expertise, and really leading the way on all of this research, none of us would be here. So my research really began with Dr. Christine with teaching me about some tissue culture and opening my eyes there. And I was able to bring that back again to Dr. Aziz's research, where he works with haploid cells, which are gamete cells, which are 1N. And so these cells are highly available as pollen cells. And through the studies with him, I discovered that through a specific stress, these cells, these haploid cells, could be matured into a plant and further um, manipulated into a fertile plant. And this is important because you start with the one end plant, and that offers a diverse assessment of it. So to facilitate this study, we had to first look at, like Dylan said, the growth characteristics of the plant, and we were able to correlate those spikelet and anther links to these microspore diameters. And by doing that, um, along with staining of the nucleuses, we are able to get a really good determination of when to harvest these plants to get these immature microspores. So here's how we basically facilitated that study. As you can see on the left is a full panicle. On the top, it contains mature microspores, and at this level, they're almost mature pollen. And at the bottom, you can harvest tetroids or tetrads. And again, we correlated the growth characteristics of the plant to this panicle and then to the diameter of the microspores. And then we confirmed it through a DAPI staining of the nucleus. A big part of our research surrounds the study of the genomics of the cell. And the reason that we have selected these cells to work with is because they lack an exide. And so we did a double confirmation of that. The first one was we stained the outer exide with aramine O. And as you can see on the bottom, from the left to the right, is a microspore maturing its exide wall. And on the right side, you can see it's very thick. And this is what doesn't allow the cell to be lysed with just animal cell kits. So we're always trying to capture cells on the left side of the spectrum where animal cell kits can lysis the kit. In addition to this, as we said, we like to do population studies. So in the top right corner, you can see a mass isolation of a heterozygous population of microspores. The smaller ones are in the unicellular stage, and the larger ones are towards the binucleus stage. All of these we can lyse with this cell kit, and as Dylan showed in his research, we're also able to dilute the solution and individually isolate these cells on a one-by-one basis and do a large population study in that manner. My personal study has taken me on a different route where I would like to convert these cells into a whole plant. So I use a different assay of plant hormones to try to promote embryogenesis from these haploid cells. And I was able to create compact and embryogenic cells from these cells. And in this regard, yeah, well, you'll have to read the paper. So these individual cell populations, as we mentioned, we like to study because, as Trinity mentioned, they are going through meiosis 1 and 2, which allows for segregation of alleles. And so we're really able to see a huge population of mutations isn't the right word, but for me, I like to look at that like SNPs. We're really able to see a huge population difference. And so we were trying to take these single cells with the Repli G kit that Dylan is using and amplifying the entire genome from these single cells, but we're also able to take a large population and amplify each one. And then we're going to look at the population studies of each one through these SSR markers that our previous master student Vinod researched and did his research on and recently graduated. So through these SSR markers, we're going to input them into the join map, and then that will allow us to see the segregation of alleles and determine how many or where the genes are located on which chromosomes.
Okay, thank you very much, Aaron, Delovan, and Trinity for telling us about your research. And as we can see that we have different aspects of research applications to improve sweet sorghum as biofuel and forage plant. And we also contacted one of the Tennessee farmer, Dr. Weimers, and he used our sweet sorghum varieties for his cattle and goats and reported back that both animals liked sweet sorghum as forage. So with that, we'll end this presentation. And if you have any question, please let us know. Thank you very much.